Hello and welcome to the 25th Vermont Grazing and Livestock Conference. I would like to take a moment to thank our sponsors. I would like to thank the National Grazing Lands Coalition, Food Animal Concerns Trust, Morrison's Custom Feeds, Rural Vermont, and the Vermont Agency of Agriculture, Food and Markets. I would also like to thank our partners. I would like to thank the United States Department of Agriculture, National Institute of Food and Agriculture, and the National Resources Conservation Service. I would also like to thank Cedar Tree Foundation and the Forrest C. and Francis H. Latner Foundation. Thank you for joining us for the 25th Grazing and Livestock Conference. The conference is brought to you by the University of Vermont Extension Center for Sustainable Agriculture and the Vermont Grass Farmers Association. Enjoy the content. I just want to introduce them both formally. So Deborah Nair is a professor, the Department of Plant and Soil Science at the University of Vermont. She's a soil ecologist. Her recent research focuses on biological communities in compost and their role in disease suppression. Deb has 30 years of experience as a researcher, educator, and graduate student mentor. Nicole Masters is an independent agroecologist, systems thinker, storyteller, and educator. Her book, For the Love of Soil, showcases examples of the principles and tools producers are using to regenerate their soil. She has over 20 years of practical and theoretical experience in regenerative agriculture on the topic of soil health. So today, Deb and Nicole both have a lot of presentations that are available on the internet. And that's not what we're going to do today. Today, we're going to take the opportunity to have Deb interview Nicole, and we're going to get a little glimpse, a unique glimpse into Nicole's work. So thank you both, and I'm going to turn it over to Deb.
Yeah, hi. Let's see, maybe just to kick it off, Nicole, how about if you just tell us maybe just a thumbnail sketch of a little bit about your life and work and how you got where you are today?
Mm-hmm. Yeah, that's always a tricky one, isn't it? How do we put this into a very short conversation? But yeah, as you can hear, I'm from New Zealand originally, and my life really these days is living on the road and working on ranches and farms in Australia and New Zealand, and predominantly in the US these days, especially with COVID. But yeah, my background is, soil ecology, soil science. But actually what got me really passionate was vermiculture and composting. So I was doing fairly large scale composting operation on the farm and creating different types of products for different types of industries. So, growing a different type of vermicast, for instance, for avocados, than maybe what we do for strawberries and what we do for pastures. But it's really hard to say. Like, my father was a pilot, so I used to travel a lot, and I think my traveling and just seeing the world from some different perspectives has given me the ability to travel and really be in people's worlds and to be able to kind of do this work without judgment. I see a lot, and I always find this question really hard because how do you talk about your life in 2 minutes? But,
no, that's great. That's helpful. It just adds that personal touch, So, I really enjoyed watching your video that was posted on there. Just for the audience today, I recognize there are a few words that might be kind of new to them, a bit familiar. So I thought I'd like you to just give a quick overview of some of the kind of sciencey kind of terms like epigenetics or quorum sensing or signaling or induced systemic resistance. Those are big words.
Yeah, and I think my interest in it, and obviously I'm not a scientist deeply working in this realm, but rather my interest in it is how does that influence what we're doing on the ranch or on the farm? And so if we think about epigenetics, we come with a genetic code. Your DNA is your DNA. no matter what, you're born with that. But epigenetics is really that argument around what shapes something. Is it the environment, or is it what you were born with? And so much of what we're seeing is epigenetic expression of the genes, which gives that gene's ability to switch on. So think of your DNA sitting there with a protein sheath that's wrapped around it. It's that protein sheath that pulls back under different environmental factors that enables that gene to express, just in really basic terms. So what does that look like? It means that a plant can switch on the ability to defend itself, or be able to switch on a gene to communicate with different types of microbiology. So that for me is some of my interest in epigenetics. If we're talking about quorum sensing, so there's quorum sensing and quorum quenching, which is the ability of a community of microbiology to reach a threshold in numbers and signal to each other with different types of chemicals that enables them to switch on that gene expression. So it might be gene expression to create biofilms, or it might be a gene expression to go virulent and create diseases. So quorum sensing is really some of the signals that microbiology and different types of animals and organisms are using to switch on. Induce and systemic responses. This is something that happens within the plant, and part of this is actually a priming system that happens in relationship with microbiology, that they send a signal, microbes respond and actually set that plant to be to induce that to a defense response. And what's interesting about that is that a lot of these responses can't happen in the plant without the presence of microbiology. What other big words do we use, Deb?
I think you hit most of the, let's see, signaling. I think, did we get that one? Yeah.
Yeah, so I talk about signaling in a really loose manner in terms of everything in soil is communicating all the time. So microbes are communicating with different types of signaling molecules or secondary metabolites. So these are things that, and two, that the plant might be producing. So flavonoids or phenolics, things that have smell, your tannins, there's all these different types of metabolites. But imagine in the soil, microbiology are wandering around. They've got no eyes and no ears. They have 100,000 receptors on the outside of their cells that are waiting for some kind of, to receive some kind of signaling. And that will tell them that there's an enemy nearby, or will tell them that there's others like them nearby. They've actually seen protozoa using these signals to hunt down nematodes, for instance. The plant is sending signals to the microbiology all the time. So there's these signals that exist in the soil, like signals to turn on a seed to germinate, for instance. So these signals are happening all the time, and there's things that we can do to influence that.
Yeah, one of those terms that I find fascinating is that quorum sensing. And the thing that it makes me think of is kind of a social organizing movement, because you have to, it's the multiple organisms. And, I had a philosopher on food ethics once asking me whether or not there's communication among plants. And I said, oh, yes. So I think between plants and plants and microbes is what you're saying. Yeah. So one thing I picked up is you talk a lot about what we call what you say is reading the landscape and soil. What does that mean to you?
Yeah, and I think this is what I'm really passionate about, training farmers and ranchers in order to do. So when you come out onto a landscape, what is it that you see growing? How is it that water is moving through that environment? What kind of biodiversity do we see? What do we see in terms of even livestock health? That all of these things are giving us clues in terms of what is happening below ground. And so I'm not saying I never get surprises, but often, I've got a sense of what's happening before you even put a shovel in the ground. Although I must say we dug holes this winter in frozen ground, and that was a surprise. What was interesting too is like the soils that were the most degraded were like solid ice, and was really hard to get the backhoe. We couldn't even dig holes. And then soils that were in really good shape didn't have that same frozen ice layer. So for me, that was kind of like, oh wow, aha. On that, like we're always surprised, but there is being able to enhance our ability to read landscape. And I think that's been the benefit of traveling all the time for me, is seeing clues.
Mm-hmm. So
Yeah.
a lot about it is soil, and you talked about the ice and stuff. Are there what other clues or cues do you pick up that you can see above ground that you automatically tells you what, it gives you an idea of what's going on below ground? What do you look for above ground?
Oh, plant
Does it tell
community is
you?
number one. Yeah,
Mm-hmm.
plant. Yeah, so certainly, I guess what people call weeds, but I would call indicator plants. Um, they're a raging clue. Things like bare
Mm-hmm.
ground, litter, the breakdown of manure. We're always getting our hands into manure and looking at, not only dung beetles, but what other species might be in there. How quickly are they breaking down? What is that dung telling you about the animal's diet? Like everything for me is a clue, I guess. Like feel like
Mm-hmm.
little Sherlock Holmes. Yeah.
Yeah, being the
Yeah.
detective. Yeah,
Yeah.
that's fun. And solving the puzzles.
Yeah. And you must see that in your work too,
Oh
right?
yeah. No, that's what I
Building.
think keeps it fun and keeps you interested, because there's always these mysteries to unravel. And
Yeah.
once I discovered kind of soil communities, you get hooked, because it's just
Yeah.
so fascinating.
It is, and it's quite funny because I don't know if we get into soil thinking that way, or I never thought, 20 odd years ago that soil was going to start being like sexy. Like people want to hear about soil and microbiology, and it's like I'm passionate about it, so why wouldn't more people be
Yeah.
passionate about it? So
Well,
yeah,
it's just like a
kind of
whole
cool.
nother universe. Most people don't even realize is existing there, right?
Yeah,
We don't even have to
yeah.
go to outer space. It's right here in our backyard.
Yeah, that's what I love about using microscopes that we can hook up to PowerPoint, is you can put someone's soil into that microscope, and then suddenly this whole invisible world becomes
Hmm.
visible. And I've had people get really emotional about seeing nematodes or even tardigrades that have come from their land, and just being so overwhelmed by, we talk about this stuff and it has that sense of kind of fairy dust, and that it's not real, but yeah, then you see it.
Yeah, that reminds me of a time I had a visitor, a photographer at one of the northern Vermont universities, come to my lab. He brought a bag of compost, and we stuck one clump under the microscope with a little water. And he spent the entire day photographing it. And,
Wow.
and he used the photos for an exhibit on living landscapes. I
Oh,
thought,
that's cool. That's really
yeah.
cool. Yeah.
So tell us why we should care about soil microbes, and also, are they all good? Are there any bad ones we need to worry about?
Yeah, well, you'd know more about that than me, but yes, there certainly are bad ones that I don't know if we need to worry about it, but I mean, I'm really, like yesterday I was driving from Billings to Big Timber, and there's a massive dust storm hitting Montana at the moment, and I think it goes all the way from the top of Montana right across South Dakota, and probably some of your listeners will be like, yeah, we've got it here too. And just being so struck by the decline of what we're seeing in soil health, how slow we are to learn as well. But this decline in soil health and the decline in our own soil, in our own microbial gut system. So human health, we're really being undermined by what we're doing to our bodies, and we're doing the same thing to the land. And then you look at how many soil microbes are actually very important for human health. And in fact, they've developed a PTSD vaccine from a type of bacterium in the soil that they're planning to give to first responders because it helps your body deal with stress. So there's so many of these interrelationships between microbiology and the soil and microbiology in us. And, really the soil was here first, and we've evolved alongside of it, breathing it, eating it. And now we're more separate from soil than we ever have been. And I think part of that and what we're seeing is that decline in human health. But yeah, we are degrading that biodiversity or the activity of microbiology in the soil systems. And who does well when that happens is our pathogens. And so now we're in these situations where maybe you don't want to be breathing in that dust or eating that soil. Whereas, 100 years ago, it would have been good to have a teaspoon of that soil. You would have got more not thinking lactobacillus, those, what do you call them?
Probiotics.
Probiotics, yeah, there'd be more probiotics in soils 120 odd years ago than what there is now. And now it'd be like, just be really careful. And we see that a lot with compost, and you'd be seeing this too, is as we degrade and make poorer quality compost, and we start to see more and more of those pathogens, or people might get exposed to legionnaires or whatever it is. Whereas someone who's worked in compost, like I've worked around compost my whole life, I'm immune to legionnaires. So I've managed to build up a system that's robust. But there are always disease organisms. It doesn't matter what it is, if that's Phytophthora, for instance, which we're having big problems with in New Zealand, a type of root rot. It is always in soil. It's just, does your soil have the ability to defend itself by having more beneficials than what it has pathogens? And I don't know if you wanted to add something to that.
Well, a couple things. One is, I think a lot of times people think of microbes, they think bad, where it's actually, wouldn't you agree, the majority of them are good and beneficial by far.
Yeah, and that's what interests me with nematodes. Like, root feeding nematodes, 95% of the research has gone into those, but only 5% of them are bad. And
Exactly.
It's like, you can see that with pharmaceuticals, and with all of our research focuses, we focus on how to kill something as opposed to, how do we create life?
Yeah.
And I think we're seeing that even with COVID right now, is instead of talking about building up our natural resilience and resistance and health and well-being and gut health, it's the reaching for the silver bullet.
Well, and the other thing I find people don't realize is that most of the antibiotics that we use in our health system originated from soil organisms. So like penicillin, ampicillin, and vancomycin.
And breathing in that soil. I'd be interested to hear your opinion on this. So we can smell geosmin at 200,000 times the strength than what a shark can smell a drop of water in an Olympic swimming pool. Why is it that, so geosmin literally translates as the odor of the earth, and part of what creates that smell is actinomycetes, what give us our antibiotics. But why is it that we can smell geosmin like that? Do you know?
I don't know, but I was going to tell you, I knew the actinomycota are the organisms that make the earthy smell that we think about, like after a rain. There's got to be some evolutionary reason for that, right? Yeah. But I'm not quite sure. That's fascinating.
I think for me, my theory is it's a reminder of where we came from. Like, that's where we evolved from. The soil microbes.
Yeah. Definitely.
That's my story. I'm sticking to it. But
let's move on to thinking about your concepts about the gene expression. And from science, we know, and you mentioned the epigenetics, the environment, affects the expression. How does this relate to or translate into agricultural practices? And, what are farmers doing that might alter gene expression in the plant?
Really good. Yeah, well, and in animals too.
That's true.
Yeah, so what's interesting, people are increasingly unhappy, I guess, with the whole genetic engineering experiment. And so actually using epigenetics is one form of plant breeding that we're starting to see gain more popularity. And the idea with this is that you would have a plant that you grow and then you stress it to different things. So extreme temperatures or waterlogging or lack of nutrition. And then you breed that plant to a healthy plant, and then you take those seeds. And then those seeds have that gene expression that's switched on to deal with those extreme conditions. And that gene expression and that change can actually last for 4 to 5 generations before that plant reverts back to its initial expression. And so we see the same thing with animals. There's quite a famous experiment where they had mice, and they exposed those mice to the smell of cherry blossom, male mice, and then every time they smelt that smell, that electrocute the poor little mouse. And so every time he smelt that smell, he gets an electrocution. Well, they bred those mice, and then for 5 generations, its progeny, if it smelt cherry blossom, would shake in fear, but not know why it was shaking. And so I was talking to some like low stress animal handling ranchers and people that are doing different things with livestock breeding, and looking at how they can really use epigenetic expression in terms of having animals that are better adapted to their environments, and looking at ways at reducing stress, because stress can have such a big impact on gene expression. So, you know, even in humans, if we're very stressed in the womb, if we don't get a loving touch as a young child, that actually affects so many genes that give us resilience to stress later on, and they don't switch on. So we don't have that ability to deal with stress later on, not because our parents didn't love us, for instance, but because our genes don't have that ability to switch on. So I think there's so much for us to explore in this.
Mm-hmm.
And a big part of it too is that relationship with microbiology.
Cool.
Yeah.
Yeah, so we'll come back to your term on signaling. You know, it's communication. I was going to say, you know, is this like a, you know, wink and a thumbs up, you know, to say, hey, it's cool. What constitutes the signal? Tell us. Well, you kind of mentioned that, but maybe who initiates this communication, and what kind of processes, you know, is it invoking?
Yeah, it's really interesting, and the processes go both ways. So, you know, plants are signaling to microbes, microbes signal to plants. A plant might say, hey, I'm under attack from a disease, and it will send a signal to specific groups of microbiology that then will respond with their own metabolites, and then the whole defense system in the plant switches on. So the plant then starts to produce more abscisic acid or different types of molecules that it'd be using for plant defense. Let's say it was under attack from an insect. That microbiology helps to support the plant in forming more of the enzyme, the proteinase enzyme. That and for me, it brings the question, that whole thing of like how undermined our soil systems are, and how bereft they are of a lot of these beneficial microbiology, or they're just not active. So the plant is signaling away and not getting any response back. Yeah, in the way that it was designed to do. And what's also interesting is, through what people have been doing with plant breeding, many of our modern cultivars no longer signal to create a relationship with mycorrhizae, or they no longer signal to communicate with protozoa, for instance. So protozoa are very important predators. They are, you know, consuming maybe 5,000 bacteria a day, and in that process they're excreting all of that nitrogen and excess nutrients back to the plant. Well, if that plant doesn't have a relationship with protozoa anymore, then it's not even telling the protozoa that it's there. Like, you should come over here, I can help you out. You know, it's all very mutualistic, a lot of these signals and a lot of these benefits.
I'm glad you brought up protozoa, because I really feel like they're the unsung heroes, and they get overlooked so often, but I think they play such a huge role, a beneficial role in our soils.
Yeah. Oh, I love them. When I see an amoeba on a slide, I get so excited. Yeah.
I know, me too. Yeah. They are amazing. And we just keep learning more. And, you know, I get fascinated. Even just the protozoa feeding on the bacteria can produce, you know, mimic a plant hormone, and then stimulate the plant to grow another lateral root, gives it a little more habitat. So, you know, just the whole, it's just, it's magic. And just the dynamic relationship that goes back and forth. So
just thinking, like, I think of this in terms of vermicast. Like, if you look at what comes out the end of a worm's butt, I call it worm butt juice, but it's like the elixir of life is what comes out the end of a worm. And you go, why is it? Like, why is it's got plant growth hormones and all sorts of secondary metabolites and phosphate solubilizing organisms and nematodes and all sorts of things come out of the back end of a worm that are perfectly set to set a plant up for health and success. And it's like, nature's just amazing. Just really, it's just incredible.
It totally is. So now that you're talking about GI tract, I got to ask you this question. In your video, you talked about the landscape being constipated. I'd love to hear what you mean by that.
Yeah, I like relating things to base instincts like bodily functions and sex. I don't know what it is, but it's, you know, a lot of soil reminds me of that. And, you know, this constipated soils, and we see a lot of this in the Midwest. You know, see a lot of this on rangeland where, you know, it's fairly low rainfall. There's not a lot of biological activity, and they're highly mineralized. And these soils, it's like they're locked up. The nutrients not going anywhere, but it's not plant available. So these are the constipated soils, as opposed to some of the soils I see in Vermont is what we call the diarrhea soils. I'm not looking at anyone in specific, but you know, there's soils where that gut function is just flushing. You know, there's nothing really holding on, and it ends up in our beautiful waterways and out at sea. Those are
now we need like a balance there, right?
Yeah.
So, yeah.
Yeah.
So that there's the nutrients are available when plants need them, and not running off and polluting our waterways, etc. Yeah, cool.
Yeah. So we do that. Yeah, we do that through restoring biological functions. So just the same in our gut, where we want to restore those tight junctions and have a microbial system that's working and digesting food, we need the same thing in our soils. And pretty much all of our modern farming practices are what disrupt our guts, just like all the modern processed food is what disrupts our guts.
Yeah, so let's expand a little bit on that, in terms of those practices, in terms of what types of practices are you referring to, and are there some of them that are worse than others, or maybe they're just have different kinds of disturbances?
Yeah, I'd say my least favorite, like my number one culprit at the top, is actually cultivation.
Mine too. I agree with you.
Yeah. So, I mean, that's despite some of these pretty heavy chemicals and things that we use, but the length of time it takes a soil to recover from a cultivation, the release of carbon, that breakdown in microbial communities. And, you know, I always look out for my wee fungi friends, because they really are the most sensitive in that ecosystem, and they really don't appreciate being chopped up.
Yeah, we're just like slicing and dicing their whole network, right?
Yeah. Yeah, and I mean, waterlogging's not great. So, you know, the property that I sold about 5 years ago, we used to get so waterlogged that soil would stink, like it would smell, like go anaerobic. It was nasty. And so we had to have a whole microbial recovery program, because if we didn't, then we'd just grow a whole lot of weeds, and you could really see that soil system become undermined. And then we start to build that sponge. So within 7 years, we had a soil that no longer would waterlog. It would just, you know, had that capacity to hold on to water. And so the practices that we use to do that is your adaptive grazing management. We had multi-species plants, we had multi-species animals. We were using microbials like the vermicast and compost, and, you know, a little bit of mineral in that process. So we really were looking at how do we address all these different aspects of building soil, and not just focusing on one part, like just the grazing or, you know, just microbials. But yeah, unfortunately, I think if you look at modern agriculture, which is the soluble fertilizers, the pesticides, the cultivation, well, maybe not cultivation, it's an old one, but not having good water management, these things are the things that all undermine that microbial community. So it's like everything that we've been doing for the last 120 odd years has really been an attack on that microbial function.
Yeah, I was thinking, yeah, as you were describing, you know, the compaction too, with heavy machinery too, and and
overgrazing. Yeah, so overgrazing would be another big culprit. And that, I mean, really, if you look around here in Montana in these big dust storms, you know, a lot of it is coming from overgrazing or cultivation.
So, yeah, and here in Vermont, we have areas that are really sandy soils, and we also have some really high clay soils. So those high clay soils, you know, I've often asked people, you know, is there any way we can reduce the cultivation? And pretty much, you know, the use of cultivation is trying to get them to, you know, get extra water, but warming up the soil so they can get into it, feeling like they have to do that. So you mentioned, a few practices that you could get around tillage, but maybe achieve some of those ecological functions, it sounds like.
Yeah, and I think if we are thinking about having warmer soils, who does that is biology. So the reason your soils are cold and wet and slow to grow is because you have a lack of functional, alive, awake biology. And we've done this on quite a few market gardens now, where it's, you know, imperative that they get that warm soil early. They do have bare soil, but we are stimulating microbiology to create that warmth instead of, yeah, like, you know, focusing on that bare soil. That didn't make sense. But yeah, I mean, you can take temperature measurements, you know, early in the season, and you want to see that you are, you know, 10, 20 degrees warmer than your neighbors as the season starts, and 10 to 20 degrees cooler in the middle of summer. That's when you know that you really are building that sponge or that apartment system for microbiology that enables air to move slowly through it. So then we have a lag, so that heat from summer actually persists through winter, and then the cool from winter persists through summer. And that's when you know that you're really getting your soil functional.
Yeah, I like thinking of that as kind of buffering capacity. You know, as you said, it's biologically driven, but it kind of helps dampen the oscillations of the extremes conditions and keeps things, more moderate and favorable for life in the soil.
Yeah.
I had another question about your signals. You know, I heard you say microbe signal and plant signal, but how far do you think these signals can reach? Because I noticed on your video you were showing string going clear across the conference room. So it made me think of distance.
Yeah, so that's thinking more of mycorrhizal penetration and the difference between mycorrhizae and non-mycorrhizae. But if we think of some of the secondary metabolites that a plant might release to signal to, so the plants are signaling as well, like they have different types of signals they might emit out a leaf when they come under attack, and they'll signal to parasitic wasps, for instance. And those signals, I think, travel about 20 meters, and then it would depend on the wind. So it's 20 meters, 20 yards. You know, and you think of some of those, like I first remembered thinking about signals when I was at university, and they were talking about the acacia grazing by giraffes, and how that acacia would send a secondary metabolite signal, and everything downwind, the other acacia would harden their leaves so that they would be less palatable. So the giraffe has learned to graze upwind because the plant hasn't received that signal to say, oi, you know, Jerry the giraffe's here. And so, you know, some of those signals travel very far. But, you know, in terms of the soil matrix, you know, it is all around gas diffusion. And so I don't know what that answer is. It's kind of an interesting one. I think for the plant, they must be signaling pretty far to think of how, like you can see this with worms. Like you've set up a worm farm. I've had worms travel like 2.5 stories up the side of my house. I was clearing out the gutters in my roof, and there were worms all in the roof. I'm like, where did you come from? And you're not thinking about how far some of these organisms will travel. And I know if I put a food source down for my worms, that food source might be like 100 feet away, and those worms will still end up all getting into it. And what they're sensing, because they don't have a sense of smell, is they're sensing vibrations. So they can feel that vibration from bacteria, which is just incredible to think about. And they can sense that change in temperature, and they'll move, same with nematodes, they'll move through these different gradients to find their food sources. So they too must be, you know, tuning into these signals.
Another signal I think about too is just carbon dioxide, just respiration. They can sense that. Yeah. So they know there's life over there.
Yes.
Or I love the concept of foracy, you know, where organisms can hitchhike on other bigger organisms as a way of getting around.
Yeah. And I mean, it's incredible to think about how fungi can have nutrients and water moving in different directions in the same place, you know, and it's like that bidirectional flow, and how plants can actually do it too, just like our spinal fluid can, that there's a bidirectional flow that we can't replicate. Like we haven't figured out how is it that fungi can do that. Is kind of cool. But yeah, how fungi are expanding and they're taking bacteria with them, or bacteria are jumping on the back of earthworms and hitching a ride to wherever they want to go. I mean, and even if you think of what's floating around in the atmosphere, I mean, if you put out a little bit of milk in a dish, it will suddenly become absolutely infested with lactobacillus because they're in the atmosphere. And we've found this with trying to do studies on compost, which shouldn't have mycorrhizae in it, or that birds bring in mycorrhizal spores and bring in protozoa on their feet, but we've also got mycorrhizal spores and protozoa probably encysted, I would imagine, in the atmosphere too.
Mm-hmm.
So yeah, I mean, the whole system's just a flurry of life.
Well, I was, you know, I'm going to go back a little bit to the distance of a signal, because once you mentioned the mycorrhizal networks under the ground, then I got thinking, you know, actually it's probably infinite, really. And I always love asking my class when I teach soil ecology, what is the largest organism in the world? And as you know, it's armillaria, a mycorrhizal fungus. So
I really want to go out to the Mailer Park in Oregon. It's on my list.
Yeah, me too. I've seen pictures, but I haven't been to it. But I don't know how many hectares it was, but it is like 65 hectares or something crazy.
No, I think it's 1,000 acres. So that would make it like,
okay, bigger than that.
400 hectares, but it's probably not that exciting, because you get there and all that is there is that the trees aren't there because they're dead. Like, that's it. But I'm going to be excited. I'm going to dig holes.
So just for the audience, one way you can recognize these too is when you see a fairy ring around a tree. That's what that is related to mycorrhizal fungi.
Yeah. And then I guess fairy rings that people see out in the field.
Mm-hmm.
For me, a lot of the time when I see those big fairy rings, they often tell me that these are potentially the constipated soils, because we find that the activity of other fungi they're not as active. And so we see that a lot in Canada. You see the most amazing, huge fairy rings. And when we go and do sampling out there, a lot of the time they are high fungal soils, but the activity is really, really low.
Okay, so that would be an above ground indicator that you're looking for.
Yeah. So I mean, I like them. They're not bad, but they're telling you that the rest of the fungal population is not as active as it could be. Yeah.
So in the Northeast, we're experiencing some intense rain events, as well as we go the other extreme in terms of more drought than we've ever seen. How do soil microbes influence these water holding capacity and help us get through the extreme weather events?
Yeah, so those microbes are vital in building that whole soil structural process. So think your bacteria make the microaggregates, so they're kind of the mortar, or not the mortar, the bricks. And then the fungi come along and make the macroaggregates, so the large crumbs. So they're building the walls pretty much, plastering everything together. And what you find then is they're building these structures. So I think of them like a huge metropolis or apartment buildings underneath the ground. And they are the ones that are increasing that gas diffusion and that water movement, and enabling water to enter and to slow, and to create more room for water to be bound in that. And what we find is my soils I go and have a look at have major compaction problems. They've lost that apartment building. They're pretty much like in these derelict car parks is basically what people are creating. And then there's no air spaces, and then there's no room for air and water to move. So it's no wonder that that we're that close to a drought all the time, even though a lot of these areas have been getting high rainfall. And New Zealand's in the same situation. So they did some sampling about 10 years ago looking at New Zealand dairy farms, and found that 78% of New Zealand dairy farms were significantly compacted. And what does that means that they're going to need more water. They need 10 times the amount of nitrogen that they used to need, because you haven't got that gas diffusion. So our job really is, how do we build all of that structure and as deep as possible? And how deep is that? Well, how deep are the deepest topsoils in the world? But, certainly it's a lot deeper than what people are currently seeing in Vermont.
You make a good point about losing the nitrogen thing in terms of if soils are waterlogged. Yeah, the problem with that denitrification and loss of nitrogen. And so, we keep putting inputs in and losing them where the biology can help hold them there and keep things functioning. I was thinking back to that issue of buffering again. Maybe too. Yeah, keeps things from getting out. Keep a balance there.
Yeah, and I think a lot about that in terms of inputs that people are using. If you are putting some nitrogen on, or you are using a herbicide, that this is the opportunity to buffer that as well. So by buffering, One of the ways I think about it really is probably in terms of chelation, which is that you're going to bind some carbon source to that nitrogen or to your herbicide so that it makes that nutrient or chemical more effective, but it's also buffering that microbiology against the negative effects. And we've one of my good friends had a study that was done on his property, and they found that once that system's built, they can deal with a bit of chemical. They actually can bioaccumulate a lot of those chemical applications if that system is robust and resilient and has that ability to buffer itself.
Cool.
Yeah,
So I just had a couple of kind of related questions, and then we'll open up the floor for some questions from the audience. So tell us a little bit kind of what's on your work horizon, you know, what you're currently working on and kind of what's inspiring you.
Oh, so many things. I have to think about what.
Give us a couple examples. Yeah.
I'm working with New Zealand Merino at the moment. So New Zealand Merino is the industry body for merino wool production in New Zealand. And they are working on producing a regenerative certification for wool that's based on outcomes. So it's not you can't do this or you must do that. Like there's no practices or anything that people are forced to do, but it looks at what are these outcomes as we start to improve soil health. And what's interesting is when they made that announcement to their members, over 50% of New Zealand high country farmers signed up. So we're talking about a pretty exciting project. Yeah, I'm working with the Intertribal Council at the moment in terms of looking at designing behavior change programs that I'm very, very excited about as well, and really looking at, building soil health on a community basis. So a lot of that community stuff gets me pretty excited. And I've got online courses that I've just released a couple, and we've got a few more coming out hopefully in the middle of the year. So they keep me pretty busy. It's really weird. Like at the moment, you know, it's winter in Montana. I'm not getting out very much, but feeling like I'm spending far too much time in front of the computer. So when we talk about stuff like this, I feel really separate from it. The most separate I think I've ever felt right now. And it's really weird. So I feel like I need to get back into the soil myself. So yeah, I need a project that's mine.
Yeah. No, that sounds very exciting.
Yeah.
Yeah. Let's back out a second and see if there's any questions coming up through the chat. I think maybe Megan was going to help us with those.
Yeah, both Jen and I are here to help be a little bit of the voice of some. There's a lot of questions.
Awesome.
I'm pretty sure we're not going to get through them all. There's
pick some top ones there.
Yeah. So what's great is they can vote them up. So, but I also just want to say, Deb and Nicole, those questions stay in that space. And so maybe at some point we can circle back around, and those that don't get asked, if you respond to them, we have that time frame throughout the whole conference for a response to those questions. So once the session is over, they stay there. So
we could answer them.
Yeah, we look into them later. So just to make people feel okay if their question doesn't get answered, because there's a lot of them, and this is a really interesting conversation. So Jen, I wanted to give you the opportunity, because with the highest rated question, I wanted it. There's a lot to it, so I'm gonna let Jen take it from here. You're muted, Jen, just so you know. Okay.
So I may not actually read this line by line, but the upshot is from a farmer, just got my soil samples back from the lab. My Morgan test says I have a 1 for phosphorus, very low, and agronomy folks tell me to apply at least 50 pounds an acre for a 4-ton dry matter per acre yield. But the regenerative tribe says, don't worry about all of it and focus on biology. So who do I really take advice from?
Great question. It's the old, all you need is biology box and all you need is mineral box. And I'm in the middle, but probably not on phosphorus applications, because we know how quickly those soluble phosphate applications can shut stuff down. If you're going to use a phosphate, then you're going to use, you really want to use a slow release phosphate. So we're not immediately telling that plant that it has all the phosphorus it could possibly need in the world, and it will stop signaling to mycorrhizae and to phosphate solubilizing bacteria. So it just, it just shuts down a lot of its carbon sink when it gets adequate phosphorus or excess phosphorus anyway. So yes, I think in the transition, and we can't give recommendations, but in transition, this is where I want to use slow release phosphorus forms. And a study was done in New Zealand, and they actually found that 1 unit of P contained in fish hydrolysate was the equivalent of 180 units contained in superphosphate. So there's different types of efficiencies with these elements. And really, when I'm looking at phosphorus, I'm going to look at the plant. So what is the phosphate level like in the plant? And the plant will tell you if it's getting phosphorus or not. They're very, very good. And so are the microbiology at releasing bound phosphorus. That's not even going to show up on your soil test, but it'll show up in the plant. So ask the plant.
So I'll just add a couple things. I mean, what you said was great, but I think it's important not to think about a nutrient in isolation of all the other nutrients. You've really got to look at the ratios and what's limiting, because so the whole story isn't just in a single one. So you have to think about it as a whole, in a holistic way. And I think another thing is a lot of the tests that are run in the labs are focusing on inorganic forms of phosphorus and not necessarily testing the range of organic forms. And I don't mean organic as in certified organic, but phosphorus that's bound in and dead and decaying material from plants or animal. And because those organic forms, those that are decaying from that, are going to be more of these slow release forms like Nicole is referring to. I mean, you can buy those. I mean, there's just a lot of different kinds of phosphorus, right?
Yeah. I just wish more agronomists were learning from people like you, Deb. And like, if you look into the literature, 90% of that P cycle is tied up in microbiology and roots and organic matter. But that's not what farmers told.
And the labs aren't testing that type of phosphorus. They're not even running the test for it. Yeah.
No.
So we're only getting part of the answer.
Yeah, that's right.
Maybe we need to go on to another one, unless there's more parts to that one.
No, great answers. Great answers. We'll just keep rolling through. We have a tie in votes for the next one. So Nicole, you were talking about a few different ways to evaluate read the landscape for landscape health. And the question is, if you could pick one, you only get to pick one keystone attribute to look at on a farm as the representation of soil regeneration, climate resilience. What would that be? What would you ask a farmer to go measure if they could only measure one thing?
Is this above or below ground? And this is measuring. See, measurement's different for me than observations. Like if I only had one observation, I'd be looking at plant indicators. So what's the community? But if it's something I'm measuring, then probably the aggregate stability. So a slaking test. What do you think?
That's exactly what I was going to say, is aggregate stability.
Yeah, that's so cool. And it's such an easy one to do. And I've got videos on the slaking test if people want to look at how to do it. But there's so much information that you can get from a slaking test, and it's such a great one to compare to your neighbors. I call it the Smug Test.
Well, the thing that's great about aggregate stability is it's an integrator. It's integrating biology and the physical structure of soil, both. You have to have both of them to make an aggregate. Without the biology, you're not going to have any aggregates.
That's so cool. Awesome. Next question. That was easy.
Glad we agree.
Thank you for resolving something. A discussion we've been having is an ongoing basis. So curious, where do you fall on bale grazing as a soil remediation and or improvement tool?
I have a process that I call the 5 M's. So looking at what is your limiting factor, is it your mindset, management, microbes, minerals, or organic matter? And what I find is, so bale grazing might not be a tool for everybody, but I really like it for a lot of these low organic matter, low biological systems. People need to be aware that they still need to manage bale grazing. I've seen a lot of very significant compaction happening from people not back fencing their bales, so ending up creating a lot of compaction issues. So you still need to manage your bales like you would for strip grazing or something. So yeah, I do like bow grazing, and I think people need to get their heads around this idea of waste and really looking at the contribution and the difference that those bow grazing situations can make, and not leaving too much behind as well. Like some people have gone too far and are leaving huge volumes, so then it probably takes 2 or 3 years before we even see growth in those areas.
I don't really have anything to add to that, but I did see another question that popped up about a book to recommend. So I reached over to my shelf and picked my book up that I'd recommend to get started. I don't know if it's going to show up, probably not, but it's by David Montgomery calling Growing a Revolution, Bringing Our Soil Back to Life. I like it because I think it's based on sound science, but it's written so that a non-scientist can follow and understand.
Yeah.
Deb, I think if you show it right in front of you, it will show up.
It's just I've got.
Yeah. Oh, no, it won't. You're right. It won't do it. So interestingly enough, we had David Montgomery a couple of years ago come and speak at our conference, which was great. So good book to pick.
Yeah, great book.
There's a question that kind of talks about the soil, the stimulation. Oh, I'm sorry, Nicole, I should let you answer that question too. Like, what book would you recommend? I didn't mean to just skip over you.
That's fine. That's fine. My book, For the Love of Soil. For the Love of Soil, Strategies to Regenerate Our Food Production Systems. Yeah, no, it's a good one.
Yeah. So there's a question about that, the stimulation, what does it look like to warm soils early in the spring? And it's tied into that tillage kind of question too. How do we reduce or eliminate the need for tillage? And then so that question, and I also wanted to tie that back to something you talked about before, which was the question of like, how long does it take for the soil, for the networks to come together after you have tilled? Like what, so it's kind of like a whole kind of, how do you eliminate tillage? And then also how long does it take for things to come back together? Recover.
It's a piece of string question, really. And the recovery time really, there's been some research that's really shown it's years and years before those networks start to recover, and it depends on what you've done. I mean, if you think of the tillage when people first broke in this country in Nebraska or Ohio, that landscape has never recovered. That microbiology has never come back. So it depends on what you're doing in terms of your management to recover that. And some of the tools I see that are very helpful are some of those seed dressings in terms of vermicast or compost or the Johnson Sioux. So really using seeds as your way of transporting microbiology and starting to get that system rebooted. But I'm seeing organic no-till starting to look like it's a possibility. I'm open to anything, but I've had lecturers that are like, there's no way you could ever be organic no-till, it's not possible. And we're seeing people here in the US get closer to that, and probably people in Vermont, with crimping, with putting in different types of covers. So yeah, I'm seeing some exciting development. And I think for some of the taller species like corn, I don't see why you couldn't do organic no-till, because you haven't got the competition. So I think we just really need to be pushing that agenda along. Like how is it possible, and what things could we do? Because we find if we change the microbial signaling, then we're not signaling for a lot of these really persistent weeds. And you look at things like, I don't know what ones you've got that are herbicide resistant, probably ryegrass. You guys don't have kochia, I don't think, but fat hen, lamb's quarters. We're seeing more and more herbicide resistance now, and those things becoming more of a problem. And what's interesting is what shuts down that pathway for herbicide resistance is actually the aromatic amino acids, but things like your fish can actually help to switch off that gene expression that creates herbicide resistance. So using products like that with your herbicides and building our soil health, then we find that we're seeing less and less of these persistent weeds and less of these production losing weeds. And that should be the goal. So if you're organic and you're finding that you're getting some of those really gnarly production losing weeds, you're sending the wrong signal to your soil.
I'm just going to, Yeah, you said a lot of great things. I was just going to second the idea of using the cover crops and the crimping. Another piece of that that's super for the biology is you always want to have plants in your soil. You don't want bare soil because it's the plants and the roots and all. That's what feeds the soil. That's where, you know, the plants are giving them their food to keep, you know, stimulate them. So that's one thing. I also wanted to say, I knew some research that it only takes one single tillage event to change the biology. And I have yet to see any system completely recover after decades. So it's, you know, it doesn't mean you can't, you know, have some healing there. You know, I just feel like it's kind of like a deep scar and you can gradually kind of improve it, but it never completely goes away. Sad, but true.
Sad, but true.
Jen, do you want to take a stab at interpreting Juan's? There's something in there and I'm trying to pull out.
Yeah, it's a comment and a question, you know, and you've been talking about this, but a little bit deeper dive would be great too. So plants are a reflection of the soils where they grow, just like cows resemble the plants that they graze. And plants that grow in healthy soils don't get sick. And plants that grow in abused or depleted soils are more vulnerable to pathogens. So how would you explain this? Would you have a general explanation as to why this is?
There's a couple of different dynamics behind this, and there's some good papers that have just come out about this. So part of it is what we call the complex amino acid or complex proteins. So we get incomplete protein formation when we have a disrupted microbial system or a disrupted plant. So there's all sorts of dynamics involved. So plant photosynthesis is obviously involved, that the plants are able to access nutrients, that they have good root penetration, that relationship with microbiology, so that they are getting the trace elements that they need for defense. So that might be manganese, or it might be copper, it might be calcium is an important one, same with boron. Are those elements being available to the plant? And then is that plant able to complex those amino acids and proteins? And what we see, and I've seen this quite a bit, just on the East Coast, is nitrates. And so those nitrates really are ringing the dinner bell for insect pests to clean it up, because in a natural environment, those nitrates are carcinogenic. They're not good for livestock, obviously. We see animals dying from excess nitrates and babies dying from nitrates in the water. So yeah, there's a whole lot of dynamics involved in that. And I've got a whole chapter on it in the book.
I want to just
So
add
there's a follow-up question too, and this could be for both of you as well. So is the use of nitrification inhibitors to improve fertilizer use efficiency starving those soil microbes, and is it worth the trade-off?
So it stops the enzyme in the specific bacterial groups from following through that nitrogen cycle. And I just think the use of those inhibitors is a slippery slope. We found it in milk in New Zealand and in the waterways, so they stopped using DCD anyway. It's the wrong, if I can say wrong mentality, it's the wrong way to really be looking at these issues, which is, one, why do you need all that nitrogen? Because you probably don't. Like, nitrogen's often not people's limiting factors. So you need to be looking at buffering that nitrogen with a carbon source, doing split applications if you need to, dealing with your trace elements, because often it is a trace element issue, and looking at compaction. If you have compacted soils, that's your problem. It's not a nitrogen problem. So no, I'm not a big supporter of those inhibitors. It's just trying to find a technological fix to a biological problem.
I wanted to add to this too a little bit that I think fits with what Nicole is saying. It's a slightly different angle. One is, you mentioned that depleted soils lead to more pathogens. Well, if you have too much fertilizer, too much nutrient, that leads to pathogens. Pathogens love nutrients, and they're going to use them better and faster and more efficiently than the beneficials. So you're actually tipping the balance towards your pathogens by adding a lot of nitrogen. So I'm with Nicole. Why are we putting so much on in the first place? Yeah. And why do we put it on and then try to turn it off? You know, why it just saves some inputs from the get-go.
It's so interesting to me. Like, I think most of the issues that we're seeing in agriculture is due to an excess of nutrient applications. not a deficiency. I mean, it's like these applications are causing most of the problems.
Mm-hmm. And it shuts off the beneficials. I think, you have too much phosphorus, the mycorrhizae aren't going to like it. It turns them off. And, why put out that effort biologically if they're lazy, so you're not going to put out the effort unless you need it to help you go out there and explore the soil for that nutrient. If you're just feeding it to them, the biology can just sit on the couch and do nothing.
Yeah, very true.
So we also have a question about how damaging is the compaction that comes from haying and from clipping?
The compaction from haying, what I see a lot really is probably more compaction from an imbalance of, like, say, alfalfa. So we see a lot of dumping of nitrogen into that soil system, which creates a lot of compaction. Yeah, I mean, I think anytime you're in a process of removing crop and then removing that potassium, so it's not being put back into the soil, then we're going to start to see issues. I mean, some of these hay fields that you go and see are some of the worst places you'll find on the ranch often. Yeah, so it's just looking at balance and, like, maybe not cutting for hay every year off those fields, maybe rotating it and having it as a winter stockpiled forage or something. I think we get stuck in doing the same thing every year instead of kind of mixing it up a bit, which might help. I don't know if you want to add anything, Deb.
Well, I was having lived on East Coast and West Coast, I was going to say, I don't think the compaction's as bad here with smaller machinery than I've seen in California. I mean, the size and weight of the machinery is one thing, but I also think about what are the conditions that you're running the machinery on, in terms of, if the soils are really wet, they're going to compact a lot easier than if it's drier. Or if you have some good organic material in your soil, that's going to help reduce the compaction. It just adds more of a spongy effect there.
Mm-hmm. Yeah. I mean, a lot of this stuff is so simple. I think it's coming back to these principles of ground cover and organic material and feeding that microbiology so that they build that structure. Yeah, we treat it like it's rocket science. It's crazy.
I was just going to add one comment about the outwinter grazing, because I did a project with, it was a conservation innovation project. We were looking at the 3 practices of, like, outwintering, and I always have to look at what the titles were. Outwintering, stockpiling, or bedded pack. And the question was whether that was detrimental to the soil quality. If that was degrading it by having all your animals out there in the winter and dealing with that. And basically the answer was no, it was not a negative impact. And one of our big metrics was aggregate stability. So it kind of calmed some of the fears and wondering if they're out there in the cold and the wet, but it's not really damaging the soil quality. So that's the good news. Interesting.
Yeah. And I think that's why it's such an important thing for people to be monitoring for this for themselves on their own properties. And look at it for yourselves. Yeah, that's interesting, guys. I mean, we find in New Zealand with the wet that we can do so much damage.
Yeah, that wet part, that's huge. That's a big challenge, right? But that getting some more sponginess in there with the soil organic matter will help. It's kind of like walking on a hard concrete floor versus one that's carpeted.
Yeah.
Okay, so we have more questions than this, but I have 2 final questions. One is swinging back to our conversation around nutrient application. And the question is, how do you overcome the pain point of transitioning from a high input system and to building and relying on your microbial community? How do you manage that transition and overcome the pain point?
Yeah, I mean, I kind of think a lot of that pain point perception came about from like 20 or 30 years ago when people were transitioning to organics and just pulling the rug out. Don't pull the rug out. There's no need for there to be any pain in this transition at all, as long as you're kind of making intelligent choices in terms of if you're reducing something, what is it that maybe needs to go in, or, trial things for yourself. But one of the best pieces of advice I got was, if you're walking through a minefield, follow in the footsteps of someone in front of you. So find someone in your local community who's done this successfully, or, work with someone that, a consultant or whatever that's really great at this transition period. So I really feel like the only risk is not doing this.
I don't really have much to add, but I was just thinking of another book that I just recently read, and I can't remember the exact author, but you know it because he's in Montana, Grain to Grain.
Oh,
Do you
yeah,
know that
Bob
book?
Quinn.
Yeah, Bob Quinn. And he explains a transition process. He was warned that that was going to be a problem, but he explains how it wasn't a problem at all in terms of how he viewed it, but he had to be very creative.
Yeah, I think the regenerative transition is not machinery intensive, it's not input intensive, it's knowledge intensive. So we need to be really investing in education and understanding some of these dynamics. And I think modern agriculture's really tried to dumb the farmer down in terms of, you don't need to understand this. The answer's 200 pounds, just put it on and use this machine. They've taken a lot of the creativity and thinking out, and regenerative agriculture is very creative thinking based, which I think is what gets people so excited about it.
Mm-hmm. Well, and the company selling you the nitrogen, sure, they want to sell you more and they want to sell you more equipment. But so I think with the biology, we're trying to encourage how to help reduce the inputs and let nature work for us. Mm-hmm. And $1 saved is $1 earned.
So speaking of creativity, our last question is a creatively oriented question. So what have you seen to be the impact of functionally integrating woody plants into pastures from a soil health and a biology perspective?
I have got a couple of clients that are doing versions of silviculture, I guess. I didn't see before they did that, but with some of my large scale operators that are, doing rootstocks or market gardening, there is a really big opportunity to do things like alley cropping, and diversifying some of those woody species in terms of, pollinators, in terms of mycorrhizal deposition. I think there's real benefits to just thinking more three-dimensionally than what we have been doing, or adopting some of the permaculture principles. It's just looking at how to do this on large-scale commercial scale, and we're seeing this popping up in Australia, which is really great, is putting in more shelter belt, more hedging, because, it's providing shelter for livestock if they're around, but also breaking up wind, and because of that biological effect. So I think integration of woody species makes a lot of sense. And I think particularly if you're looking at alternative income streams. I know what you want to add, Deb.
Yeah, well, you hit on part of it, but I was just going to add that roots again, you're going to get some deeper roots, and they're really going to help you with the infiltration. So to, keep that water moving through, and also they can help you avoid extra heavy, lots of ponding of water too, because they're going to evaporate that water. So they're good for that part too. So you're going to get those roots, more life in the soil. They're going to have a different depth than your grasses. So, having the mixture just adds more variety.
Well, thank you both very much. And on behalf of the Vermont Grazing Livestock Conference Committee, we're very excited that you were able to join us both. There's lots more questions that have sort of ended up in our Q&A. So if you do get the chance to circle around and answer some of those, I'm sure folks would really appreciate that. And if folks have taken a look, we've got a couple of polls just to check in and see what you learned today and how things have been going. I encourage you to do that, continue the conversation and chat. And we look forward to seeing you all later tonight if you can make it for the Farmer Storytelling Hour at 5 o'clock. And if not, just keep talking to each other just as you are doing through the community and the HUVA interaction. It's been fantastic. So thank you again, Deb, and thank you again very much, Nicole, for joining us.
Thank you so much. Thanks for that great interview, Deb. Great work.
Yeah, it was fun. It's been nice to get acquainted, Nicole, and I look forward to following your work.
Yes, you as well. Brilliant. All right. Thanks, everybody. Have a great
All right.
conference.
Thanks.
Bye.